Good day everyone, welcome to this video. We are the Group 4 reporter in GE106. My name is Mo Kevin L. Cordova from BSc Shem1C. Our topic is about genetically modified organism or GMO. In this video, we are going to discuss about the definition of the GMO, the production of the GMO, the principal objective of production, the existing models of GMO, the genetic engineering versus traditional breeding in GMO, and also the pros and cons of the GMO. And to proceed with our topic, I will be starting off with what is GMO. So GMO is a genetically modified organism. It is an organism whose genetic material has been altered using genetic engineering techniques. The exact definition of a genetically modified organism and what constitutes genetic engineering may vary, with the most common being an organism altered in a way that does not occur naturally or by mating or a natural recombination. A genetically modified organism is an animal, plant, or microbe whose DNA has been altered using a genetic engineering techniques. For 1,000 years, humans have been using breeding methods to modify organisms such as corn, cattle, and even dogs that have been selected bred over generation to have a certain desired trait. And now, let's proceed to the production of the GMO.
Production of genetically modified organism. Genetic modification involves the insertion of deletion of genes. When genes are inserted, they usually come from a different species, which is a form of horizontal gene transfer. In nature, this can occur when exogenous DNA penetrates the cell membrane for any reason. To do this artificially may require attaching the genes to a virus or just physically inserting the extra DNA into the nucleus of the intended host with a very small syringe with very small particles fired from a gene. Agrobacteriocin, ability to transfer genetic material to plants, or the ability of lentiviruses to transfer gene to animal cells are natural examples of gene transfer.
Principal objective of production. The basic principle for producing a GMO was to add new genetic material into an organism genome. This is called genetic engineering and was made possible through the discovery of DNA and the creation of the first recombinant DNA molecule by Paul Berg in 1972.
Existing models. Although planning crucial part of the system development process, it is often neglected by project managers. The problem being addressed by this paper is that for inadequate models for planning the requirements capture and analysis RAA of a software development project. It is stressed that there is need for a new model because existing models give inaccurate, inconsistent, or unreliable predictions. Additionally, they are based on either inaccurate variables or variables that can be measured at the beginning of the development process. Finally, existing models do not support the planning of individual stages of the development process but only to make prediction about project development process as a whole. This paper examines existing models and provides evidence about their inaccuracy and lack of accuracy, and then introduces a new model and presents the approach followed for its development.
Genetic engineering versus traditional breeding. With traditional breeding, plants often exchange large, unregulated chunks of their genomes. This can lead to both useful and unwanted traits in the offspring. Sometimes, these unwanted traits can be unsafe. One example would be potato varieties made using conventional plant breeding that inadvertently produce excessive levels of natural occurring glycocolloids. These glycocolloids cause gastrointestinal, circulatory, neurological, and dermatological problems associated with alkaloid poisoning. Breeders sometimes have to cross many plants over multiple generations to produce the desired trait. GM techniques allow new traits to be introduced one at a time, without complication from extra genes and extensive crossbreeding. GM techniques also allow traits from different organisms to be applied, such as a pest.
Pros and cons of genetically modified organisms. The pros of GMO crops are that they may contain more nutrients, are grown with fewer pesticides, and are usually cheaper than their non-GMO counterparts. The cons of GMO foods are that they may cause allergic reactions because of their altered DNA, and they may increase antibiotic resistance.
This is the report of Group 4. Thank you for listening.